In this talk today, I want to try to explain to you when, where, and how thalidomide acted to cause the malformations that you've had through your lives. And my contribution today is going to be a mixture of history and science. Along the way, I want to introduce you to some of the people who helped me behind the scenes, because you can't do this sort of thing on your own. My involvement began when your compensation became available in the early 1970s. Many handicapped children queued up at that time for the money, along with the proven thalidomiders. Some mothers had no thalidomide history, and it was hard to decide who should be compensated and who should not. In Sydney, I was sent a bundle of X-ray films, and I was asked whether there was any feature in the X-ray films that identified a thalidomide from a non-thalidomide defect. Parents of 5 Australian children, whose X-rays I had in my collection, had kept the prescription or the bottle of tablets, and those 5 children were already accepted medico-legally as prima facie thalidomide cases. So I studied the X-ray films of these 5 cases carefully to identify the features of proven thalidomide damage. It was immediately obvious that a pattern of malformations was repeated in these 5 children. For instance, 4 out of the 5 children were missing part or all of the radius in the forearm and the thumb on both sides. Reduction of the radius and thumb had been the most common defect in the upper limb in all the big German and British series. So the question was this: when, where and how did the drug do this to a normal embryo? The when question had already been answered by doctors in Germany. Based on histories of hundreds of German children, Novak and Lenz in Germany discovered when the drug acted, and this was between 21 and 42 days after conception. And they called that the thalidomide-sensitive period. Within this period, arm defects happened early, from day 24. But before this, indeed, in the very earliest part of the period, there were deformities of the head and neck, and the eyes and the ears. And later than the arm defects came the leg defects, always in a head-to-tail sequence. So the question boiled down to: where and how did the drug act to cause absence of the radius? I searched the literature for an explanation of this strange pattern of bone loss. There was no pathology to be found. Pathologists had searched the bones and the cartilage in vain for the target tissue. They found that the cartilage and bone cells were normal, with no disease process here to match these X-ray images. The longitudinal distribution of defects was not characteristic of bone disease. Bone diseases, for instance, do not jump across joints, nor do bone diseases delete long strips or rows of bone, leaving normal adjacent or normal residual bony remnants. These skeletal defects did not obey the laws of bone disease. Therefore, you had to think of another tissue that was damaged. Although the most dramatic feature was loss or reduction in the skeleton, absence of bone pathology in the skeleton meant that this was not a primary disease of bone, but of some other system. So the next question looms up: if it's not a primary bone disease, what else could it be? Well, both blood vessels and nerves supply bones. Thalidomide might attack either of these, with secondary damage to the bones. That would explain what we're looking at. First of all, the blood vessels. There were at least 4 reasons why a disease of blood vessels was unlikely. First of all, thalidomide did not cause any vascular disorders in adults, but it did cause sensory peripheral nerve damage in adults who used it as a sedative before it was used for morning sickness. Secondly, blood vessels in the limb buds of the embryos are rich with interconnecting vessels. It's a lacy network, rather like a mosquito net. There are almost infinite numbers of detours and alternative routes for blood to flow through around any blocked vessel, and this would make it impossible to delete a row of bones through lack of blood supply. Thirdly, there are arterial arcades, we call them, in the palm of the hand that give off small arteries to the digits. It's anatomically unlikely for one or two digital arteries to be injured without damaging others branching off the same arcade. To achieve reduction of the radial bone and absence of the thumb, one would have to damage several tiny branch arteries at the same time without damaging their main stem. Multiple sites of damage were unlikely to give similar results time after time. Fourthly, at the embryonic age of 24 days, when thalidomide causes arm defects, the limb bud and its arteries do not yet exist. This is powerful evidence against it being anything arterial. Let's look at the timelines. Here we have the sensitive period of the embryo, and we have an arrow here at day 24 where thalidomide comes in and causes arm defects. But we've also got a column here that shows that the limb bud for the arm first appears at day 28, 4 days after thalidomide's done its damage. Wherever thalidomide acts, we can say it does not act in the limb bud because the arm bud and its arteries do not yet exist at 24 days of gestation. The thalidomide molecule itself has a half-life of 3 hours. It is all broken down by 6 hours. It does not wait around for 4 days. It's vanished by 6 hours after taking. Now the arm buds have just appeared in this 28-day human embryo. The same fact has been recorded in apes and monkeys given thalidomide. The arm bud is not present when thalidomide causes the arm defects. Dieter Neubert of Berlin maintains from his 40 years of studying thalidomide in primates that the drug acts within the torso of the embryo, deep to the site of the future limb bud. So, does this drug act on the blood vessels? No, it doesn't. So let's look at the nerves. The central nervous system is the control room. It's the main dynamo of the whole nervous system. And the central nervous system consists of the brain and the spinal cord, both of which are derived from the neural tube in the embryo. But as well as the central nervous system, we've got an enormous ramification of nerve fibres going out to the edge of the body. And this is the peripheral nervous system. It's the wiring, if you like. It's that huge network of wires that reach out to all parts of the body, carrying both sensory and autonomic messages between the control room of the central nervous system and the environment outside the organism. The peripheral nervous system is derived not from the neural tube, but from an adjacent tissue called the neural crest of the embryo. These peripheral nerves develop sequentially from head to tail, 2 at a time, that is, one on the right, one on the left, and segment by segment as it works its maturation down the body. The cell body of each nerve, with the nucleus inside it, is always located in the ganglion that sits beside the spinal cord, and the nerves are just long strings of cytoplasm pulled out from that cell and called axons, and they penetrate into all corners of the body. Now the neural crest is busily forming from day 18, which is 10 days before any limb bud appears. So from the start, nerves are precocious. They're more advanced than the limbs and any other organs that they supply. And they're developing right and left sides, pair by pair. The nerves, therefore, are there when thalidomide strikes at day 24 to cause the arm defects, in contrast with the vessels that are not there. From day 18 to 21, the embryonic nervous system is actively developing down the back of the embryo 10 days before the arm bud appears. The sensory nervous system is busily forming from head to tail during this time. Cranial nerves appear first, then the spinal segmental nerves, the cervical, then the thoracic, then the lumbar, and finally the sacral nerves, all in due sequence and separated by short intervals in time. Thus, each pair of emerging segmental nerves is separated by this small time interval from the level above and the level below. This means that at any moment, one segment is going to be more vulnerable than the one above or the one below or any other segment. And it's a sort of chronological keyhole into which a foreign neurotoxin can slide. Just one segment at a time in this peripheral nerve system can thus be damaged. Now, the nerve supply to the internal organs also comes from the neural crest. These nerve cells actually migrate into ancillary ganglia and then migrate down to actually surround the organs that they're there to supply. And while they're doing this, they're multiplying and they're sprouting axons. So from day 21 of embryonic life, neural crest cells in the ganglia sprout axons that extend out with the growing embryo, sliding between the stem cells in the intercellular spaces, setting up the wiring system as the body parts evolve. At day 28, nerve centres are formed and are ready to innervate the limb bud, which is not yet present. At day 37, the limb bud is a primitive paddle full of simple stem cells. But look at how sophisticated the peripheral nerves are. Developmentally, the nerves are racing ahead of the limb buds. Does thalidomide target nerves? Yes, it does. Thalidomide poisoned adult sensory nerves. It caused profound sensory nerve damage of the axonal degenerative type in people who used it as a sedative prior to its use as an antiemetic for morning sickness. It should therefore come as no surprise that thalidomide poisons sensory nerves in the embryo, the neural crest, the parent of the sensory and the autonomic nerves. The nerves are present and active before the limbs exist, as we've just seen. Thirdly, nerves characteristically maintain an end-organ relationship with the structures they supply. Blood vessels don't do this. They tend to wander and deviate from their original destination. So we look now at what we call the dermatomes of the body. And here we have what I call the pancake man. The peripheral nerves supply the skin of the body in orderly strips, and each strip is called a dermatome. And each dermatome belongs to one spinal segmental nerve. They're formed horizontally in the embryo, so you've got to sit a man in this ungainly position in order to see how they've evolved. And they evolve, if you like a stack of pancakes. There's a neat stack in the chest and the abdomen, where you can see through the ribs and peripheral structures what the bands of the segments are like. But where you've got the arms and legs emerging from the torso, this stack gets disordered and looks untidy and distorted as the limbs grow out. But the segmental order is still there underneath. Now, for instance, here is the dermatome of the sixth cervical nerve from the neck, and it encompasses the skin of the thumb and the skin of this side of the forearm overlying the radius. The sixth cervical dermatome is coloured black. Now, if you took a sharp knife and you cut away this area of skin plus the bones underneath, you would end up with absent radius and thumb. In other words, The thalidomide disease seems to fit the distribution of segmental sensory nerves. But here's a problem. I've run into it headlong. They say in the textbooks that there are no nerves in limb buds. I was instantly worried that my observations may have gone astray. And the more I read about birth defects, the more worried I became. My conclusions about nerves being the underlying tissue that caused the defects is in total conflict with accepted dogma. Perhaps I was building a house of cards. I checked and I rechecked my reasoning, and I couldn't find any errors. But had I gone wrong in interpreting the X-rays, I wondered? I needed to check that my reading of the X-rays was correct. So in 1972, I took my material to a trusted friend and authority in Bristol. Here he is, Professor Howard Middlemiss, and he was also the president of the College of Radiologists of the UK at that stage. Now, Sir Howard, I knew, had studied and written about sensory nerve damage in leprosy, and I knew that he had a collection of X-rays of African lepers. And let me assure you that the only connection between thalidomide and leprosy is that they both injure sensory nerves and eliminate bones. I went to see Professor Middlemiss in Bristol in order to compare X-rays of the bones and joints of my Australian thalidomiders with X-rays of his African lepers. I knew that he would tell me if my observations or deductions had gone wrong. Together we went through the body from head to foot. Bone by bone and joint by joint, we compared my thalidomiders from Australia and his lepers from Africa. Bone by bone, joint by joint, and we found the same X-ray changes in these two groups. He told me that my observations were correct. He also said this was the first time that radiology had identified a disease process before the pathology was known. So I said to him, but the embryology textbooks say there are no nerves in limb buds. He replied that the embryology textbooks were wrong and that my deductions were correct. He said that this was the most important thing I'd ever done, and I must continue the research no matter what. I took a paper called Thalidomide and Congenital Charcot's Joints to the Lancet a few months later, and I had a similar response from the deputy editor, Dr. Ian Munro. He told me that before he became a medical journalist, he had been a diagnostic radiologist at Guy's Hospital, so he knew exactly what I was seeing in the X-rays, and he fast-tracked my article into print. After that publication, I had hundreds of requests for reprints. But the Lancet editor also received several angry letters from outraged embryologists and developmental biologists whose dogma I had contradicted. I was branded a heretic from that point on in some quarters. Now here's Professor Lowenthal, who was Professor of Surgery at the University of Sydney, and radiology came under surgery within the academic world in those days. Professor Lowenthal really liked my theory of neural crest injury. He insisted that I should leave my job as a clinical radiologist at the teaching hospital and transfer to Sydney University Department of Surgery to do full-time research. I argued with him. I said I wanted to keep my job and I wanted to do research part-time. He argued back that you can't do research part-time, certainly not when you've got clinical responsibilities as well. It had to be full-time or not at all. So, reluctantly, my life changed from that of a clinician to that of a researcher. This meant you could really address questions front on. Now, the radiology clearly indicated that there was some connection between sensory nerves and limb growth. On my study leave in Britain in 1972, I spent a few weeks in the library of the Royal Society of Medicine, where I stumbled upon a treasure trove in the literature. And this was a eureka moment. I knew exactly how Archimedes felt when he leapt out of the bath and ran naked down the street shouting, "Eureka, I have it!" Fortunately, I was fully clothed and sitting in a silent medical library, but nevertheless bursting with the same excitement. What I had found was this: a series of papers that showed that sensory nerves stimulate the growth of limbs, a property called neurotrophism. Neuro for nerves, trophism for growth. A group of developmental biologists in Cleveland, Ohio, were investigating neurotrophism using amphibian models, that is, newts and axolotls which can regrow their limbs when they're cut off. Newts have a very important primitive ability that higher animals have lost during evolution. They have this ability to regrow tails and legs if they get bitten off by a predator. In its natural habitat of a pond, an injured newt just hides away in the weeds for about 6 weeks until a new leg or arm has grown from the stump of the old one. Biologists say that these amphibia are adults who retain the embryonic property of limb growth. So studying the regeneration of newt limbs should shed light on how embryos grow limbs. The leader of this team, shown here, is Professor Marcus Singer, who had PhDs in 3 subjects in anatomy, neurology and biology. I became a regular member of his group, and you could say one of his disciples, and I used to join their research seminars every summer for several years while I learned how newts grow new limbs. Now, he had shown in one of his first experiments that if he damaged the sensory nerve to the stump, the limb regrowth wouldn't happen. By examining the operation sites in these newts, Singer found that he had not done what he thought he'd done. In fact, he had crushed or avulsed the ganglion itself. He had destroyed the nerve cell bodies. He concluded that limbs will not grow without a sensory nerve supply. A man called Wallace from England, a biologist, did a beautiful study in Singer's laboratory and showed that mitosis or cell division requires to be triggered by a sensory nerve supply. Nerve triggers newt limb growth by stimulating cell division at the amputation site. So the little bulge growing out of that is full of nerves and full of mitotic figures. So here is the essence of neurotrophism. The relationship between nerves and growth is that sensory nerves trigger cell division. Nerves stimulate mitosis of other cells, that is, they make them divide. They say, divide, divide, divide. It's a simple message. Thus, nerves initiate growth. Next, we can see how many cell divisions make up a heap of cells. Nerves always act like the officers in the army, and the stem cells act like simple soldiers. If the nerve keeps barking the order, divide, 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 stem cells keep on dividing. And this creates a pile of cells, such as the early limb bud of the embryo. So, nerves initiate the first stage of formation of the limb bud. And, left undisturbed, neurotrophism will create the perfect number of cells in the limb bud for future differentiation to take place and form the perfect limb. And now we look at the formation of limbs in the embryo, where nerve axons sprout in the embryo, where nerves generate more stem cells downstream and thereby increase the number of cells in the limb bud, and they make the limb bud grow. So now we can understand how normal limbs grow. Their cells are driven to divide by sensory nerves, And these nerves are present, penetrating into the spaces between the cells. Now if we introduce thalidomide and allow it to slide into the chronological keyhole and severely damage one segmental nerve, we've got a failure of that nerve to grow. The failure of the C6 dermatome to grow leaves you with a defect in that area of nerve supply. This shows the sclerotome maps of arms and legs. Now, the segmental sensory nerve supply of the bones of the limbs was mapped out by two Americans in 1944, Inman and Saunders, and they said that a band of the skeleton was supplied by one segmental nerve, and that skeletal tissue supplied by that nerve was called its sclerotome. In other words, it's like a dermatome, which refers to skin, and this is the skeletal equivalent or the bony equivalent of the dermatome. In the arm, only the sclerotomes of the cervical nerves 6, 7 and 8 reach the hand. When I first showed these maps to Martin Johnson of the Thalidomide Trust, he exclaimed, "But there are the malformations." Then he explained to me that as an airman in a previous life, he had learned map reading. The deformities just jumped at him out of the sclerotome maps. Next series of slides show a damaged embryonic nerve which ceases to function so that its neurotrophic action stops, so cell division downstream ceases in that area of nerve supply, and the growth of the bone in that region stops, so there's no bony sclerotome in that region. Watch the black band and the blue band. In the case of damage to C6, no C6 sclerotome is present, so no black band forms in the limb bud, so you get absence of the radius and the thumb. In the case of excessive damage, which hits C5 as well as C6, no black or blue bands form. This means the humerus is going to be shorter because it's had the black and blue bits removed. And that brings the arm and the hand up towards the shoulder, which in other words is phocomelia. If we look at the leg, absence of the fourth lumbar sclerotome causes the initial defect in the thalidomide series in the legs, which was absence of the tibia from the bottom end up. And the next slide shows 3 Australian cases of absence of the tibia due to thalidomide. Now, I needed to test the sclerotome maps in a large collection of X-rays of thalidomide children. So I sought out a German professor of orthopaedics, Hans Willert, whose thalidomide papers I had read and very much appreciated. It was obvious that we both thought in terms of anatomy. He still had a collection of X-rays of over 200 German thalidomiders on which he and Professor Henkel had defined the pattern of thalidomide embryopathy in the skeleton, the pattern of what thalidomide does to it. Now this slide shows the pattern of dysmelia. I took the sclerotome maps to Professor Willert in Göttingen, Germany. He had the material and I had the method, and we put these two together. We worked through his collection of X-rays, case by case, and we established that the sclerotome maps explained the disordered anatomy in thalidomide embryopathy. We found 80% fitted very well, 15% were what you might call a fair fit, and 5% fitted poorly. Biologically, this is a very high correlation rate. So, in our neurology laboratory at Sydney University, my team established that there were nerves in limb buds before any differentiation into other tissues. Here we have the electron microscopy done by Dr. John Cameron showing nerve fibres within the limb bud. We also looked for pathology in the nerves of rabbit babies exposed to thalidomide. And here we show that all the rabbit pups exposed to thalidomide have a reduction in size and number of their axon populations. In answer to the question of when, where and how thalidomide acts, we can say that the when question was answered by Lentz and Novak, who established the sensitive period of day 21 to 42 post-conception. That's when thalidomide acted, just when a woman's thinking she might be pregnant. Within that period, they showed how it caused arm defects from day 24, head and neck problems before this, and lower leg defects later. So when is already answered by Lentz and Novak. Where did it act? It acts on the neural crest. And how does it act? It injures the developing neural crest, either their cells or their neurons or their axons, and it halts their neurotrophic function, so they fail to trigger cell division downstream, and without that you get no limb growth in that band of nerve supply. This is a Goya picture which has some messages for us. It's a charcoal sketch by Goya from 18th century Madrid, long before the thalidomide molecule was invented, and yet here we have what's typically a thalidomide a tetrafocomelic baby in the lap of its mother. We don't know what agent injured this infant when it was an embryo, but we can suggest that a sensory nerve poison acted on the same final common pathway as thalidomide to generate the same limb defects. In other words, this baby has had a neural crest injury within that sensitive period, but not due to thalidomide. Goya is telling us that thalidomide is not unique in causing these malformations. It's not the only neurotoxin in our environment. But nevertheless, thalidomide has been responsible for revealing neurotrophism as a previously unrecognised mechanism of embryonic development. It's an exquisitely sensitive mechanism that's vulnerable to environmental toxins.